Thank you, Professor Krysko. Actually, your research reminds us that even in death of cells, of course, there can be a renewal, that understanding how cells die may one day help us save lives.
Yes, in order to survive in our body, the cells should properly die.
Yes.
That's correct.
Thank you very much.
Thank you.
One, two, three. Hi everyone. Today we have again another special guest, Dmitri Krysko, an associate professor, head of the Cell Death Investigation and Therapy, or CDIT, laboratory at the Department of Human Structure and Repair at Ghent University. He's also an elected member of the Royal Academy of Medicine of Belgium since 2023. So I'm very honored to have you here. Thank you for being here.
Thank you.
So how did your fascination with cell death begin, and what led you to focus your career on it?
Well, actually, my interest in cell death goes back to my medical studies when I was a student of Saratov State Medical University in Russia. And I was actually, I won mobility stage, so I went to United States of America to one of the top universities in States, University of North Carolina in Chapel Hill. And during this stay, I got fascinated with research, and specifically with cell death research. And that was actually the starting point of my interest in cell death. And since then, I'm still working on cell death, so more than 25 years. So I started as a PhD student in Belgium in one of the top groups led by Professor Katarina Deerda. And she's also my promoter of my PhD thesis. And actually, yeah, it was a very good luck that I had the opportunity to meet her. And actually, it was really a fortune to meet her in Belgium. And also, I have to say that I did part of my PhD in one of the top institutions in Belgium, led by Professor Peter van den Nambele. This is VIB. And I did PhD actually between two labs, Professor Katarina Deerda and Peter van den Nambele. So that was my starting point from medical studies in Russia to Ghent, to Belgium.
Yeah, indeed. You did have a very rich international background, and so I just want to know what were the main reasons or motivations behind founding the Cell Death Investigation and Therapy, or CDIT, laboratory at Ghent University?
Well, actually, I was fascinated with cell death because if you, at the moment when I was a medical student in 1997, it was just one cell death modality, apoptotic cell death modality. And it translates from Greeks as falling leaves from the tree in autumn. So meaning that it's very physiological process of cell death. But since then, we have more than 12 cell death modalities, regulated cell death modalities. And I would like to ask you, do you know how many well, first of all, do you think that you're alive now?
I would like to think so.
But I will challenge this opinion because do you know how many cells die per minute, for example, in our body? Just can you guess?
Millions?
Millions, that's correct. So in our body, 1 to 3 million of cells die each minute. And if we calculate this to the year basis, more than 75 kilograms of our body weight get recycled. Imagine 75 kilograms, it's average person weight, get recycled during 1 year. So we have renewal of our organs, we have renewal of our systems, and that's is very fascinating. Therefore, well, I this subject keeps my interest, and I'm still interested in this subject. But of course, I moved from cell death research now to cancer immunotherapy, and this is what we are doing in my laboratory. We studying and we try to develop novel cancer immunotherapeutics for cancer, different types of cancer, and one of them is glioblastoma.
Mhm. Actually, that's a very interesting point that you mentioned. I would like to challenge what you think. Do you think you are alive, that every second millions of cell is dying? In the meaning that we are regenerating our cells every millisecond or every second. So that's very interesting. Also, I came across just about this statement from earlier interviews. A better understanding of mechanisms of cell death on the long run may lead to more efficient cancer immunotherapy. Can you elaborate this statement?
Yes, I can elaborate because, well, first of all, I would like to say that in our organism there is a balance between cell division and cell death. There is a balance. For example, there are several diseases like myocardial infarction, stroke, autoimmune diseases, and in those diseases we have actually too much of cell death. So the balance is towards too much of cell death, and it means that people and scientists develop strategies to block cell death in these diseases in order to rescue patients. But apparently, the lack of cell death is neither beneficial. So there are diseases in which lack of cell death can be part of the pathology, like, for example, in cancer. And we know that in cancer, there is mutations which actually make cells resistant. Resistance meaning that they are not capable to die. And that's actually the problem, because then if you treat patients with specific therapy, either chemotherapy or radiotherapy, and then those cancer cells are not capable to die, and that results in relapses, and it makes the therapy is less efficient. But if we can understand the mechanisms of cell death, then we can regulate, and we can influence, we can direct the cells to cell death. And that's why the understanding of mechanisms of cell death is very important in order to be able to manipulate, if you wish, will cell death process, either to block it, for example, in conditions when there is too high cell death, like myocardial infarction, stroke, or stimulate cell death. And this is what we are doing in cancer. So in cancer therapy, you need to have efficient cell death of cancer cells.
Mhm. Your lab also focuses on immunogenic cell death, ICD. For our listeners, could you explain what immunogenic cell death is and why it matters?
Well, immunogenic cell death, it's a very nice concept which arose around 2005, and actually it was proposed by the group of Professor Guido Kromer in France, in Paris, and we also contributed to the development of this cell death concept. Actually, what it states that actually the most types of the therapy, anti-cancer therapy, just kill cells, just eliminate them, and it becomes clear that it's not enough. because that, as I mentioned before, cells develop resistance and they are not capable to die anymore. So this strategy will not work 100% in the whole tumor. So you will have cells which will survive, and that will make a problem. But immunogenic cell death implies that in addition to cell death induction, you will also activate the host, the patient immune system in order to help to combat against cancerous cells. And that's very advantageous. So it's actually twofold. From one hand, you kill cancerous cells in a specific way, immunogenic way, and then those cells induce anti-cancer immunity. And this anti-cancer immunity is very crucial for long-term survival of patients, for reducing relapse, because immune cells of the patient will run in the body and will kill those remaining cells which are not killed by chemotherapy. Therefore, induction of immunogenic cell death is very beneficial, and not all types of anti-cancer therapy can do this. But actually, this is the aim, one of the aim of my laboratory. We develop novel strategies to kill cancerous cells on immunogenic way. Hello, Maria.
Hello.
I want to introduce Marii Saviuk. Marii Saviuk is a third-year PhD student, and Maria joined the CDIT lab in December 2022. Actually, Maria graduated from one of the top universities in Russia, Lobachevsky State Medical University in Nizhny Novgorod. But I know Maria already for more than 4 years, or maybe even 5 years, because you did bachelor work in our lab and also master student work, master thesis work. And actually, it's really a good fortune to have Maria in the lab, because Maria is very hardworking. Maria can do very elegantly intracranial tumor models in mice, where you need to do kind of a microsurgery in mice. And Maria is doing this very successful. And Maria is a driver, actually, and the first author on this manuscript in Nature Communication, and the driver of the project. And I will give a word to Maria.
Thank you.
What first drew you to the study of cell death and cancer?
Well, I started my studies when I was a bachelor student, and I started with neuroscience, and it was amazing. But also I had a little bit of cell death in my bachelor studies. And then I came here for a small stage for 2 months, and here it was completely cell death. No, a little bit of neurology as well, because it was glioblastoma. It's a brain tumor, so still neurology, but also a lot of cell death. How cells dies, how we should to kill them, and I was amazed. And then I came back for my master stage, and I stayed here for PhD.
Can you tell us more about your current PhD research?
Now I work with glioblastoma. Glioblastoma, it's a brain tumor, and it's extremely dangerous. The median survival rate of glioblastoma is 1 year. It means that half of the patients will die in 1 year after the diagnosis with the standard treatment. The standard treatment was made 20 years ago, and it's approved a little bit, but not that crucial, and survival rates are not improved. So now we want to make a new treatment for glioblastoma to make the life of the patients longer and better. And in my research, I work with glioblastoma. Now we work with mostly murine cells, so we inject glioblastoma cells to the brain, and then we are trying to treat it. And how we do it? We have our immune system, and actually we can use immune system to kill tumor cells. So we can teach our immune system how to fight cancer. And we are trying to do that. We use dendritic cell vaccines. And if you know nothing about immune system, I will explain you briefly. We have an innate immune system, and we have immune system which we can train. And we use the second one. So we have dendritic cells. Dendritic cells, it's kind of the cells which we can give them some tumor cells, and then we can use those dendritic cells. They will be trained, and they will teach another immune cells how to kill tumor cells. So we teach immune system how to kill tumor cells, and that's my PhD study.
So, what is regulated cell death and what role does it play in immunogenic cell death research?
There are many cells in our organism, and like you know, the middle person with a weight of 75 kilos have within 1 year changed 75 kilos of cells. So every day millions and billions sometimes of the cells dies in our organism. And they don't die just randomly. Normally each cell knows when it should to die. And when it dies, normally nothing really dangerous happens in the organism because the cells know that they have to die and another cells come, clean it, and it's fine. And it's normal regulated cell death. But sometimes we have immunogenic cell death. It's a type of regulated cell death, but cells then not just die. They die with fireworks and fanfares, and those fireworks we call damage-associated molecular patterns. And when they die with fireworks, then immune cells can see those dead cells and they can use them to be activated. So regulated cell death happens often, always basically in our organism, and immunogenic cell death, it happens, and after immunogenic cell death, immune system can be really active.
Can you tell us about the gliomas and treatments that currently exist?
Well, I already told a little bit about gliomas. It's dangerous tumors, and the standard treatment now is surgical resection in combination with radiotherapy and chemotherapy. And it was made 20 years ago, and there are not many options for the patients. And there are some new clinical trials for glioblastoma, which using immunotherapy. Immunotherapy means that we want to teach immune system how to fight cancer. And there are different ways of immunotherapies. It can be antigen vaccine. So normally when we use immunotherapy in our context, we use a sample from patient and then we modify it somehow, and then we inject it back to the patient, and then the immune system from the patient become active. There are also some different ways of immunotherapy. It can be immune checkpoint inhibitors. Probably many people who knows a little bit about breast cancer knows about immune checkpoint inhibitors because it's quite often used for breast cancer. And surprisingly, it was tested for glioblastoma as well in mice, and it's a really fun story for Science Fail Night conference that it worked amazing in glioblastoma in mice, and many papers were published. They started clinical trial and it's completely failed because in glioblastoma cell lines they don't have this marker. They have it in murine cells but not in human cells. And now they're trying to incorporate it with another immune treatments, and apparently it should be efficient as well, but not in the way as it's efficient for breast cancer.
Oh, you are in your third year, and I see that you are very passionate with your research. So, what advice would you give for incoming PhDs entering this field?
You should be confident about what you're doing and what you're going to do, but in the same time you should be able to change your idea, because when I came we had one idea but then we had to change it because it failed. And don't be afraid about negative results. It's a part of science, and to be honest, I think 85% of all my results are negative. And it's normal for science, because you have some ideas and then it can be successful and it can fail. And it's normal that some results can be failed and some results can be negative, some experiments can be negative, but still use those data because you cannot make a conclusion based only on positive results, only on positive experiments. You also have to have some negative, and don't be afraid and don't cry because of them.
Your lab also uses 3D cancer spheroids to mimic real tumor environments. What have these models revealed that traditional cell cultures cannot?
Well, indeed, most of the research is done on 2D cultures, meaning that you seed cells in one monolayer on a plastic. And this very, of course, easy to study. So the cells very easy to manipulate with them, to induce cell death, to follow their behavior. But this is not fully reflecting in vivo situation, because in vivo you have cell-cell contacts, you have other cell types, which actually create tumor microenvironment. And generating spheroids actually allow to mimic in vitro situation what we have in vivo. So this is more relevant because you have cell-cell contacts, you have also gradient of hypoxia, which indicates less of oxygen in the tissue. You have also you can also generate spheroids with multiple cell types, and this helps actually to mimic and to develop and to test therapies in vitro. And those therapies would be more efficient in vivo because they are tested in a very relevant, clinically relevant situation using 3D spheroids.
That's very interesting. Could you also explain us what are anti-cancer vaccines and is it around the corner?
Well, yes, anti-cancer vaccines. Actually, there are two types of anti-cancer vaccines. There are preventive anti-cancer vaccines and therapeutic anti-cancer vaccines. So there are already existing on the clinical market, and patients receive such vaccines, prophylactic vaccines. One of them is human papilloma virus vaccines. So it's and hepatitis B vaccines. So these vaccines actually done preventively in order to prevent development of cancers. But also there are therapeutic vaccines, and they are still under development. Meaning, if a patient will develop a cancer, then we need to treat this patient while he has a tumor or cancer disease. So those types of two main cancer vaccines. And in our laboratory, actually, we develop cancer vaccines, therapeutic cancer vaccines, and we focus on glioblastoma. Glioblastoma is one of the most frequently met tumors of central nervous system. So it's in the brain and it's very deadly. So despite the therapy, extensive therapy like chemotherapy, radiotherapy, there is 90% of patients will have a relapse after 6 or 7 months, and actually 5-year survival is less than 7%. It's very deadly tumor, and therefore we started this project about 3.5 years ago, and we wanted to develop cancer vaccines based on immunogenic cell death, more specifically on ferroptotic cell death. So ferroptosis actually is one of regulated cell death modalities, which is driven by lipid peroxidation, and it is sensitive on iron and sensitive on it's sensitive on reactive oxygen species. And actually, what we found that if we kill ferroptotic, if we kill glioma cells by ferroptosis, if we induce ferroptosis in glioma cells, and then if we actually co-culture those dying cells and we generate lysates from them, and then we put those lysates on dendritic cells. Actually, those are antigen-presenting cells, which induce anti-tumor immunity, and those then we have AGC vaccines. So DC vaccines consisting of dendritic cells and therapeutically dying glioma cells. And we found that these vaccines are very efficient in mice models, in orthotopic intracranial mouse models. And actually the theory behind of that we want to take biopsies, and biopsies can be taken, this is piece of the tumors, which can be taken from the patient during the surgery. And then those biopsies or piece of the tissue, tumor tissue, can be put in the culture, and we can induce ferroptotic cell death in those tumor-derived cells. And then we can generate lysates in a specific way and put on also patient-derived monocytes. So we take from the same patient a blood sample, and we, using specific technology, expand monocytes and differentiate into dendritic cells. And then we take, so actually it's from the same patient, we have monocytes, dendritic cells, and a biopsy, and we put together. In this way, we educate dendritic cells of a patient in vitro. They will acquire certain characteristics, and then we can inject back those dendritic cells into the same person. So we are speaking about personalized therapy. It's a very expensive therapy, but it's personalized therapy because we obtain material from the same person, and then we inject this therapy back to the same person. And we hope that now we are validating our studies on human primary materials. So we have collaboration with neurosurgeons here in Ghent University Hospital. And we hope that in the coming 1 year or two, we can validate these findings on human material. And if it turns to be positive, then we will be ready to run first clinical trial to test this immunotherapy and to treat patients, first group of patients, and to see efficacy of those vaccines in patient.
Mhm. That's actually very interesting explanation. And you also mentioned a lot about patients that these anti-cancer vaccines are given to the patients with the cancer, right? So what are the main challenges in translating the findings in controlled studies into real cancer treatments?
Well, the main challenge that, of course, we also use, people and scientists use mouse models, and sometimes, well, we use also orthotopic intracranial glioma mouse models, which resemble very closely clinical situation in the patient. But the problem is that, we use quite artificial cancer cells, and also, there is a huge heterogeneity of the tumor in patients, and therefore it makes it's difficult to translate the findings which are obtained in the lab, in the experimental settings, to the patient. But we have a very good hope because we have used intracranial therapeutic models. What means intracranial? So we actually using stereotactic equipment, we inject first viable cells into the brain of the mice, and then after a certain period, like a week or so, we start the therapy with such educated dendritic cells. And actually, this is a very clinically relevant situation because we have development of the tumor in the brain in a similar way as in the patient. And we have obtained in these therapeutic settings, and we actually uncovered the mechanisms of this action in vitro and in vivo. And we believe that this therapy will be also efficient in glioblastoma patients. But of course, first we need to validate this in vitro on human patient biopsies, and later on, if positive, we will run a clinical trial.
Thank you very much for your insights. Actually, you are one of the leading researchers in this domain, and it's nice to have you today at the podcast as well, your lab. To just conclude the episode, I have one general question, if you don't mind.
Yes, sure.
So, what advice would you give to young scientists entering this area?
Well, it's a very good question, actually. I think for science, you need to have a lot of energy. You need to be inspired by the subject, and this should be actually your main hobby, not a work but a hobby. And if you will do this will a pleasure, and you will enjoy doing this, then you will have a success. And the second important element of success is mobility. So you need to have a good mobility. As in my case, I had the mobility to United States, University of North Carolina. And this mobility will actually expand your horizons, scientific horizons. It will also help to create a network, scientific network. And of course, the third element is reading scientific articles, and dreaming about possible mechanisms and possible new therapies.
Thank you, Professor Krysko. Actually, your research reminds us that even in death of cells, of course, there can be a renewal, that understanding how cells die may one day help us save lives.
Yes, in order to survive in our body, the cells should properly die.
Yes.
That's correct.
Thank you very much.
Thank you.